Hello, everybody. I'd like to thank you all for being here. My name is Amanda Clark, and I am the director of the UCLA Center for Reproductive Science, Health, and Education. The vision for our center is for a future in which reproductive policies and care are based on rigorous science, and access to this care is more equitable. As reproductive health and justice have become part of the national conversation, a pressing need has emerged to ensure that reproductive science research, education, and outreach are keeping pace with the public need for accurate, fact-based information and continued scientific innovation that will improve the lives of people and their families. In 2023, we launched this one-of-a-kind center at UCLA to serve both academic and public interests in communicating the science of reproductive health, the connection to reproductive justice, and to build a pipeline for future careers in this area. The center hosts career development activities, offers undergraduate courses, holds a monthly seminar series, and funds research fellowships that promote cross-disciplinary engagement in reproductive science, sexual health, and maternal health. The center's major public engagement event is this Distinguished Speaker Series, which is why you're here today. Infertility is a disease that affects around 17% of the adult population worldwide and is of significant public health concern. Improving infertility treatments is a challenge for U.S. scientists due to the need to navigate a patchwork of policy and regulatory barriers that were created over a decade ago. This has stifled innovations to improve technologies such as IVF. Professor Magdalena Zernicka-Goetz is successfully navigating these challenges, but it has not been easy for her or for any of us that work in this space, including investigators at UCLA like Catherine Plath. We are delighted that she is joining us today to talk about her research. This event would not be possible without the generous support of Tom Rando and the UCLA Broad Stem Cell Research Center, Jeff Long and the Department of Molecular Cell and Developmental Biology, and Grace Hong and the Barbra Streisand Center in the Division of Social Sciences. I would also like to give a special thanks to Stephanie Kiso-Ido, Stacey Antonou, and Nushig Karpanian for their work at putting this event together today. With the Center's social science work in mind, I am delighted to introduce Hannah Landecker, Associate Dean of Undergraduate Education in the Life Sciences, Professor in the Institute for Society and Genetics, Professor in the Department of Sociology, and Co-Director of the Center. Professor Landecker will provide a short historical perspective on the topic of human embryos and human developmental biology and introduce our distinguished speaker. Thank you, Hannah.
Hello, everyone. Because we are here today to talk about the early human embryo, I wanted to kick things off extremely briefly by situating us historically in the long arc of a 150-year or more struggle to generate systematic visualization and experimental techniques to understand what is going on in these very tiny bodies and very early stages of life. Some of the earliest efforts were these now classic drawings of Ernst Haeckel from the 1870s. These were highly controversial because of their depictions of the similarity of human embryos to those of other species. These drawings were also immediately fiercely critiqued for their scientific accuracy. The depictions, of course, are highly related to how we know what we know about embryos. And Haeckel's compatriots thought that they were highly limited by their two-dimensionality and the interpretative hand of the artist, that is Haeckel himself. In response, other investigators experimented with glass plate photography, followed by drawing, and the reconstruction from cross-sections of the embryo in wax models, making a model dissection of the wax embryo to reproduce its interior structure in 3 dimensions. But what of the 4th dimension of time? And I think possibly we will hear about facing these problems in the contemporary today. This interesting lithograph adopts Haeckel's efforts to use colors to show development over time and what happens to the different layers of the developing embryo. The historian Nick Hopwood explains that this image was actually a parody of Haeckel's drawings. It was meant to accompany a song set to the tune of Nach Italien, to Italy, that visits the attractions of each diagram as a destination until, dizzy from following the lines, the singer retires to the pub for beer and verst in the bottom right of the diagram, just in case you were wondering if 19th century embryologists had a sense of humor. Yet, of course, let's make this play. The achievement of accessing continuous views of development in space and time for early human embryos was and is extremely difficult. The very first ever time-lapse film of embryonic development that you see before you here was made in 1908 with a fertilized sea urchin egg, exactly because they were relatively large and easy to keep alive in seawater. Julius Rees, the maker of the film, wrote that he made it because the empirical observation of events in time was necessary to the theoretical understanding of development. But very few people could go to marine biological stations and see such sites for themselves. As a result, he lamented, cell theory was something true that a student must just believe without being able to convince himself of it. The experience of seeing the film, he thought, was the impression that one has the living, developing egg before one. So today, we are in for a contemporary version of these revelatory experiences and the problems and questions and challenges of visualizing and studying early development in time and space. It is a great pleasure to introduce to you Magdalena Zernicka-Goetz, who is the Bren Professor of Biology and Biological Engineering at Caltech and Professor of Mammalian Development and Stem Cells at the University of Cambridge. Spanning the last 25 years, research from her lab has broken new ground in studies of human embryo post-implantation development, cell fate specification in mouse and human embryos, and the creation of multiple stem cell-based embryo models to understand the fundamental principles and mechanisms that regulate cell identity, pluripotency, and embryoplasticity. All, of course, moving towards the goals that Amanda mentioned, improving assisted reproductive technologies and helping elucidate the causes of early pregnancy failure, providing insights into the developmental origins of disease. Within an impressive and very long list of papers and accomplishments, I just want to mention a few key recent ones. In 2023, in the journal Nature, Dr. Zernicka-Goetz and colleagues published the first model of the human post-implantation embryo, significantly accelerating research into the causes of early pregnancy loss. Also in 2023, she was awarded the Ogawa Yamanaka Stem Cell Prize from the Gladstone Institute. This prize recognizes groundbreaking work in translational regenerative medicine using reprogrammed cells. She was selected for the prize based on her fundamental studies leading to the technology of embryo models. Thus, please join me in welcoming our speaker today to hear about the science of human embryos and embryo models. Welcome.
Thank you so much, Amanda and Hannah and Tom and everybody who came to listen to my talk. This is really a great pleasure to be here. I have such admiration, not only to topic, but also to UCLA as a whole. That it's really a honor, and I'm so pleased I'm a neighbor now. I'm going to try to switch to the different computer, and I hope that it will go without any problems. Let's see. Yes. Okay, fantastic. Yes.
Thank you so much. I have this two parts. So, so what I would like to do today is to tell you about the work we have been doing in the lab now for quite a while, that really tries to understand how we can discover some of the basic principles, even if not all, some of the basics ones that make it possible for our embryos to develop so successfully to create us and other mammals. And today, because I'm giving a talk in the center that is so much aiming on improving reproduction and also understand the beginning of our own life, I would like to focus exclusively on human embryo development. So hopefully I would manage it within the half of an hour. If not, I would just cut one story short. If you have any questions during the talk, I also invite you to ask them, but I know that Amanda told me that at the end of the talk, we will have also a possibility to discuss some of the implications of our work also from the ethical perspective. So I would like to tell you why I'm very interested in these early stages of development and principles. And the major reason is that I would like really to understand how it is possible that an egg, very simple cell, that looks very similar in mouse and human and many other mammals, can create such incredibly complex and beautiful individuals within a relatively short period of time. And when you think about this early process of creation, actually all of those individuals, despite of being different from each other, start their life in exactly the same way. So what it is about these early stages of life that unites us? The second major reason for so this is sort of basic interest, like curiosity driven motivation. The second basic interest really is due to the fact that majority of the pregnancies fail. So if you look at this pregnancy iceberg, you see that 60% of those failures happen within the first 2 weeks of our life. And this is the time when actually we still do not know we are pregnant. So what it is about embryo that cannot really fulfill these early stages of its development? Actually, when I'm thinking about it, I would think about that most lives end before they even begin. If we think about the beginning of the life is the birth. So to be able to address the question of, how we are building ourselves and why so often this building is inaccurate, that leads to abortion, we have been studying mouse embryo development for now more than 25 years and trying to understand when and how cells become different to form these three lineages in these three different colors and how these three different types of cells talk to each other to build something much more complex, but which you can sort of see the head and the rest of the body and the tail. So you can see how the adult with anterior posterior axis becomes specified. So only recently, maybe 10 years ago, we started also to look at human embryo development directly. And as you can see, we know very little about early stages of human embryo development. And despite the fact that we actually can culture mouse and human embryos in vitro, and I will tell you the story that we just recently uncovered, like very surprising result that relate to these early stages of development to allow us to put some colors on this diagram below too. The later stages of human development were equally enigmatic, and this is because we were not able to culture human embryo beyond the blastocyst stage. Now, I put those colors on these drawings. I shouldn't have, but a few years ago, we found a little bit more about what happens at the time, and also there is an access to embryos recovered from the body of the mother. So the access was possible in 60s when we were able to see some of the in vivo developing embryos being harvested at different stages, very early stages of pregnancy. So we know a little bit more about this, some of the milestones that happened at the time, but how they are connected with each other was enigmatic. Okay, so these are the three questions that I would like to address today. How does the embryo establish its first tissues to create diversity? So how we go from one cell to many and how those cells know what they should make. Then what happens when those cells interact with each other? So when we established the first three tissues, then the cells start to interact with each other to build embryonic body. And finally, if I am allowed, meaning like if my time will be not totally off, I will tell you a little bit about environmental factors that impact embryo development that we can learn by building embryo models. So we don't have to study them directly, how they impact real human embryos, but we can study their impact on embryo models. Okay. So cell identity, so how cells know what to make. So in many model organisms, the dogma is, and actually based on many, many experiments, that there is some early asymmetry either in the egg after fertilization or even sometimes before fertilization. And if you have such an asymmetric egg, when you divide it into smaller cells, those cells will have different information, and there will be some information of which type of cells they should build when they build the embryo proper later. But in mammals, the dogma was saying that it is not the case because it is very difficult to identify any determinant localized at any specific places within the egg or even early cleavage stage embryos. So it was thought for a very long time that in mammals, all of the cells are identical, they are totipotent until quite late in development in comparison to other species, until the morula stage embryos in which you have some inside and some outside cells. So those inside cells will be the one that will make inner cell mass, and this is the inner cell mass that will create embryonic stem cells for the future body, while outside cells will make the placenta. Now, this, when I was a student, so I know many faces that I see here, I think are student faces. So when I was you, this is the view that I had, and we all the textbooks and also my professors, they were teaching me that mammals are different than other model systems. And I didn't ever question it. I was not planning to question it. But when I set up my own research group in Cambridge, I wanted to develop life markers for cells. So essentially, GFP didn't exist at the time. You perhaps find it very surprising, but it didn't. So I wanted to use something like lymph-fluorescent protein that just was discovered, was produced by Aquaria Victoria, and thought that maybe I can paint cells in the early embryo to look at the fate of those cells in the system that is very plastic. We knew about plasticity of mammalian development. And those painting experiments with GFP and many other markers led me to find something extremely, at the time, controversial and unexpected. So we found that already at the 2-cell stage in the mouse, there start to be differences between those cells. So one cell will be more totipotent than the other. And I still would call them totipotent because they have a huge plasticity, but there will be a degree of variability in the extent of how good they are to make the body versus placenta. So I'm not going to go through many of those studies, but you see, we published and other groups published many papers over years to actually find what are the factors in the egg that might be their positioned asymmetrically, how it affects this molecular asymmetry, what are the epigenetic marks that become, and epigenetic players, I should say, that are involved at this 2-cell stage, how they affect cleavage pattern from 2 to 4. And finally, how those events affects which cells go inside to make the inner cell mass. So all of those experiments finally explain a mystery in studies of Anne McLaren, who was one of the amazing woman embryologists. When I was growing up, I was sharing office with her, and I thought that I was so honored by being able to share her office because she was much older than me, much more established. I was just setting up my research group. That I still remember those conversations. One of the dilemmas that we discussed is that when you take 2-cell stage embryo and cleave it into 2 halves at the 2-cell stage, only one will make a mouse and the second would most of the time fail. But it was always interpreted that these cells are identical, and it's just the experimental difficulties of keeping 2 halves alive under microscope. So we now know that there are these early heterogeneities in cell fate that established themselves very early. As I've mentioned, was extremely controversial for many years, and that's why it took so many papers to really find out why and how. And this is because system is plastic, so it's very difficult to make a definitive experiment, remove something, and then the rest will fail. But the question is whether the same is true in humans. So, okay, mouse, we have some early cell fate specification bias. What about human embryos? It was actually not possible to address this question in human embryos very easily. And this is because when human embryos are donated for research, so they're not created for research, donated for research, they would be donated at the blastocyst stage. So this is the stage of development that already you have inside cells and outside cells. So, you wouldn't be able to sort of track it back to the earlier stages of development. So because this issue was controversial, costed me a lot of emotional strength for many years when I was growing up, I thought that it would be really, really nice if we can one day check whether this is also true in humans or not. But I couldn't have access to the embryos at 2-cell stage or zygote stage. So it was only when I came here to live in California, I found a clinic, IVF clinic, that actually had very old human zygotes stored at that stage of development. And luckily, they were donated for research, and they were donated for our research. So I would like to show you the human embryo developing in culture. So this is how we look, all of us look like. You see, you have 2 pronuclei. At one point, they will disappear just to make a spindle, and then this egg will divide into smaller and smaller cells until it forms blastocysts. In humans, this takes 6 days. So we know that our bodies derive from this inside group of cells, and we know that this group of cells is generated by inside cells within the morula stage embryos based on the mouse experiment. But the question is, can we also track back this human embryo development to even earlier stages? And if so, what it is about those cells that maybe become different from each other. So how cells lose their totipotency for the first time. So to address this question, we took 2-cell stage embryo, human embryo, and we injected at random 1 blastomer at the 2-cell stage with synthetic transcripts encoding GFP using this method that I established for mouse, which is non-invasive labeling method. So you inject those cells, but they don't know they were injected. So this was important because the fate is plastic at the time. And we analyzed many of those embryos, all of them that were donated to us, and found a very surprising result because the bias here was even stronger than in the mouse. So Trophectodem lineage was derived from both blastomers in majority of the embryos, but inner cell mass was derived predominantly from 1 cell at the 2-cell stage. So I didn't tell you that because I want to complicate it, but actually inner cell mass have 2 types of characters. Only 1 type of inner cell mass cells called the epiblast will make the body. The second also will form extraembryonic structure, yolk sac. So we asked the question whether all inner cell mass cells are biased by carried out more experiments, and we found out that actually this is the epiblast, so the group of stem cells that will make the body that is derived predominantly from 1 blastomer, the 2-cell stage. The second lineage, like Trophectodem, can be contributed by both. So this was amazing result, but of course we wanted to understand how it happens. So, what is the mechanism behind it? So how those cells become different from each other. So for that, we had an access to another set of actually early cleavage stage human embryos, and we were able not only to label them with GFP or not, but we were able to add this fluorescent dyes that will mark either membrane or nucleus because they have affinity to those structures. We don't have to inject them because at the later stages it will be very invasive to inject every single cell and will take a long time, and we want not to perturb human embryo. So we just add these dyes to the culture medium to label individual cells so we can segment them and we can track them. We can make those lineage trees and identify the pattern of asymmetric and symmetric divisions. So asymmetric divisions will be the one that will push 1 cell inside to make the pluripotent progeny of inner cell mass, and asymmetric divisions will make both cells going outside. So when we analyze all of this data, we found that these asymmetric divisions at the 8 to 16 cell stage transition are very rare. But when they come, they really impact composition of the inner cell mass. Of course, at that moment, we exhausted all of the human embryos that were donated to our research at zygote stage. So we went to really look a little bit further into it to collaborate with IVF clinics that some of the IVF clinics do movies of human embryo development under very simple scope. So it's not confocal scope. We can't label those embryos for fluorescent dyes unless we have them in the lab setting. Those embryos are really the ones that will be transferred to the parents, to the future parents. But we found out that many of those clinics that do image human embryos, they image them, they often analyze 1 single plane, And just they look at the gross morphological changes. Do they divide to two-cell stage, whether the cells look more or less the same, and so on and so forth. But actually, often that image is collected at multiple planes, or at least if you ask them, they can collect the image at multiple planes. So despite the fact that they are not fluorescent, because obviously they will be real humans, so they would not be labeled, not able to label those cells, we were able just to track the cells a little bit from 8 to 16 cells they transition to look at these division patterns. And what we discovered is that the cell that divides first at the two-cell stage is the one that will take the first asymmetric division. And this will be the cell that will decide to make the body. I mean, the cell doesn't decide, but you know what I mean. And the other cell will make, will focus on making the placenta. So actually, then I wish to say that my son and another 17-year-old teenager came to my lab over Christmas when I was in Cambridge, and they start to analyze those movies. And my son discovered by chance that this blastomer that developed first is actually bigger than the other. So it's really his micro publication in Micro Publication Journal. It's a one figure in the journal. I highly recommend this journal for tiny little projects that one day you might have if their funding is important. Then you can have one figure in which you explain your findings. So that's what happened to him. I didn't know about this microjournal before he did this discovery, but here we are. So he contributed a little bit scientifically to our life. So I was happy. I was happy that kept him busy through Christmas. So essentially what I wanted to say in the summary, so what we now know is really this shows this new, leads to the new paradigm that the first sulfate decision in mammals are actually asymmetric. The first division of the human zygote is asymmetric in size, and it's also asymmetric in setting up dynamics that the first of those two blastomers will divide. So one of the two blastomers divide first and will take a lead, and this will be the blastomers that will contribute to the future body. Okay, so we now know this, and a very simple story, but dream of my life, finally realized I know that, but of course we don't know really mechanistically what set up this asymmetry. So I will tell you a little bit what we plan to do to address this question. Actually, I have forgotten to show you this slide, so it just popped in, so I've forgotten about it, that I have it. I wanted to mention the number of papers over the recent years when they analyzed biopsies of dead bodies, and found out, to surprise, that majority of organs, if not all of the organs, come indeed from one cell at the two-cell stage. And this was like, so I think now we understand why it is so. And this was the backtracking through the somatic mutations. Okay, so what we would like to do right now is to, of course, ask the question how the cells become different from each other. And I will tell you a little bit about the story of how we identify alpha and beta blastomer by single-cell proteomics. We would like to be able to split a two-cell embryo into two, as was done for the mouse, and check whether indeed one can make a normal-looking blastocyst and the other will be not totipotent, that will make many trophoblasts. And finally, what it is within these inside cells that will make them different from each other to follow two different paths. So I'm running a little bit out of time, so I am just going to maybe just deliver bottom line for those experiments, and you can imagine what we have done here. So we took again two-cell stage embryo. We did it for mouse and human. Remove zona pellucida, separated into two cells, and use single-cell proteomics, because in the past we used RNA-seq data and we found many transcripts being differential expressed in those cells. But it was partially due to the noise, so it's very difficult to find out which of those transcripts really matters. And when we had this lineage tracing studies results first, we realized that the difference in those cells might be not because of RNA, but because of protein, as it became obvious before zygotic genome activation. I didn't make this point, but actually 8-cell stage is the stage when human embryo initiates zygotic genome activation process. So therefore, in the mouse, it's 2-cell stage, so we wouldn't know, but from human, we suspected that it could be maternal set of proteins. So we separated those cells and collaborated with Nikolai Slavov lab in Boston, who developed a beautiful protocol for single cell proteomics. And when we analyzed those data, we found out that there are many proteins that are present on to different extent at the two-cell stage. And it's not like that bigger or smaller cells have more or less of these proteins. We were able to identify a group of proteins that will be much more visible in one cell and a group that will be less visible in one cell and opposite in the other. So we were able to identify sort of alpha and beta character of those blastomers. And those proteins, they are important for transport, for degradation, and for translation. So there are all these processes at the beginning of our life that really, really matter of how to regulate developmental processes. So then, what is it really important, this alpha-beta character? So the experiment that we have done, and we did it in the mouse, is to take two-cell stage embryo and now separate it into two cells and culture one cell to the blastocyst stage to check whether it will form normal blastocysts or not. And the second cell was sent to Boston from a lab in Cambridge to check protein profile of the cells to know whether it's alpha or beta. Then we will know whether those other sister is alpha or beta. So this is what we did. So at the blastocyst stage, we counted the number of inside cells. We know from earlier work in the lab that if you have more than 4 epiblast cells by the time of implantation, you have a chance to survive. But if the number is less than 4, then those embryos will be aborted. So we were able to look at all of those embryos and now find their identity. And we found that beta cells have a higher developmental potential than alpha. So these are the really, truly pluripotent cells. Okay, now it doesn't mean that the story is over, because now you can ask the question like we did several years ago in this review, wanting to trace the origin of sulfate. So now we know that these differences are set up at the two-cell stage, but still is the question, what is setting them up? What it is it a maternal protein that become asymmetric localized? And if so, is it in relation to the fertilization or maturation of the egg? And we don't know the answer, but we know the answer will be complex because the clues at the time that exist are very subtle. So this is number 1 question. And number 2 relates to the interactions between cells at the blastocyst stage. So I've told you now one story that helped us understand how these 3 different lineages become established at the blastocyst stage. So I've told you that this red group of cells is derived predominantly from one cell at the 2-cell stage. But this is just the first week of our life. What happens in the second week? So in the second week, we have this process that we call implantation. It's the time when the blastocyst cannot survive anymore outside the body of the mother, but have to invade the body of the mother. And this is the very first time when embryo starts to grow and the cells start to properly interact with each other. And we wanted to know this process, but was not possible to culture embryos beyond implantation in vitro, either for the mouse or for the human. And we know that up to the blastocyst stage, mouse and human embryos look very similar to each other. But from the time of implantation, they start to differ. So in mouse, we have this X-cylindrical structure, so the structure elongates. And in humans, we form this epiblast disc, so the structure is flat. This red group of cells, the epiblast, is only of one type in the mouse before gastrulation. Gastrulation starts on one side soon later. And in humans, we form 2 types of epiblast cells. So one of them will be devoted to making the body and the second to making the amnion. So again, how this split happens also was not clear because it does not happen at this stage of life in mouse. So to be able to address this transition from pre to post-implantation fate, actually, yes, I've forgotten to tell that, as you see, there are some differences, but in both cases, the group of epiblast cells have to polarize to form this unilateral unicellular epithelium. So we established 2 approaches to be able to address this question. Now, I feel like I have echo from time to time when I speak. Do you feel the same? Do you hear the same? Tiny amount. I don't know whether if I put it further away, do you hear still me? Yeah? Okay. All right. So to be able to go further, we had to establish the new culture system that would be much richer than pre-implantation culture system, that would allow those embryos to grow naturally and form those two different structures in mouse and human. And the lessons that we learn through seeing how embryos develop in natural circumstances, despite the ex vivo, allow us to make this first embryonic stem cell derived models of embryo for mouse and human. And I will tell you a little bit about both, focusing on human. So like with all of the experiments, we always start first with the mouse. So this is the culture system that we establish and check whether we can take mouse blastocysts and culture it to this excylindrical structure. And we found that finally we found there was a lot of work conditions that allow us to do this. And in this first paper with that system, we devoted our attention to this anterior group of cells shown here in green, which develops in one of these two extraembryonic structures. And this group of cells is extremely important in deciding where we put our head. So it's believed that this is the group of cells that actually breaks embryo symmetry for the very first time at post-implantation stages. So breaking symmetry, pre-implantation was two-cell asymmetry, and now we are talking about a more complex situation in which we have to decide which cells will make a head. So we were able to use this system to track the origins of this anterior signaling center all the way back to the pre-implantation blastocyst. Luckily for us, the same culture system that we set up for mouse when we tried in humans, it worked. And here is one of our first human embryos cultured through this peri-implantation stages. Here in white, you see this group of pluripotent cells derived from one blastomer, two-cell stage, OCT4, they express OCT4, it's a transcription factor that marks this group of epiblast cells. And you can see how those cells proliferate and form this structure. We were able also to identify the same signaling center in humans. It's shown here in green, again marking Cerberus, so it's one of the genes that the center expresses to protect this part of the epiblast for the head formation. And here's just one of the more recent human embryos that I'm showing, again, this Cerberus signaling center here to protect this future head. So you see that it's still far away from making a head, but you can still the sign of symmetry breaking actually very soon after embryo implants. Okay, so implantation barrier is overcome, and I am not going to show you many examples of mouse and human embryos that we studied to really understand those interactions, because I would like to really now focus exclusively on this approach number 2 that is interesting because provides new opportunities to study human development without the need to use real human embryos. And this is to build the stem cell based embryo models. So as I've mentioned, it's only really by finding out how mouse and human embryo transit from those group of pluripotent cells to form sort of beginning of the cavity and to form this first epithelium, gave us the cues of how we might be able to take mouse ES cells and human ES cells and mimic these processes in culture. So now we can really study this. We are embedding those mouse or human embryonic stem cells in the matrogel that mimic the presence to certain extents of these extraembryonic tissues that are not present here. And we can now look at the process of how cells exit from naive state of pluripotency to prime, which pathways are important for this transition, how it relates to expression of the important protein, podocalaxin, that allows the lumen to form. So we are still studying this model system. It's very, very basic, very, very simple model system, but very influential for many of the aspects that we would like to study, such as how different states of pluripotency regulates different morphogenetic events that have to be fulfilled at that stage. But of course, it would be really much better if we could not just recreate only the epiblast of the embryo in culture, but whole embryo. And for that, we need to add extraembryonic tissues. So you remember that we have these 3 different characters at the blastocyst stage. So now we are mimicking development of red cells, epiblast, but can we add stem cells for primitive endoderm and trophoectoderm And we know that these stem cells can be maintained and cultured in vitro. Many groups established them, particularly Janet Rosand group for trophoblast and Zen cells, and of course Martin Evans and Gail Martin for ES cells, but they're all growing in different culture conditions. So by now we have established 7 different models how we can put those different stem cells together to mimic embryo-like structure. And I would like to show you just model number 3. This model combines all 3 types of stem cells. We published it in this paper in 2018, and this was work of Berna Sozen and Gianluca Amadei in the lab. We found conditions that allow us to replace extracellular matrix by adding TS and Zen cells. We add the cells to the agrowell dish. And now we can look at the process of self-assembly to ask how cells know where to go. And we found that it's mediated by cadherin code. Then we can look at the process of lumen formation, not only in the epiblast compartment, but also in trophoectoderm-derived compartment extraembryonic ectoderm compartment derived from trophoectoderm. We can look how those 2 lumens come together to form proamniotic cavity. We can look at specification of this anterior signaling center and its migration towards one side to set up apiaxis. So this model is absolutely incredible. It's extremely powerful, but it's mouse model. And the question is, can we do something like that? Oh, okay, for humans. So before I would go there, I would like to show that this model also formed primordial germ cells. I wanted to show it to you because I know that Amanda's lab might be here, and Amanda's beautiful work really help us to understand how PGCs are formed in human context. So this is really very important question to understand how those cells form and what are the potential of those cells. So in this mouse model, we see their formation on the boundary between different compartments. This is the formation of the head in this mouse embryo model with the most anterior aspect, which is quite unique for models. We form the heart, and we have this neural tube closure, and we form also somites. So it's really pretty good-looking embryo. Not all of those embryos derived from stem cells look like that, so we still call them embryo-like structures rather than embryos. But they are pretty powerful because now you can manipulate different individual lineages and look at the processes of the mechanism that leads to their specification. But can we do it for humans? So this would be even more important because despite of the fact that having mouse model help us to understand the principles of human development, of course, mouse and humans are not the same, particularly after implantation. And why we have access to mouse embryos after implantation, it's hard to study at the time, much harder than pre-implantation, we have access to them. For human embryos, obviously they are donated to research at the blastocyst stage, but you can imagine not many human embryos will be donated to research. So actually genetic perturbations in that system to understand the true deeply mechanistic, to provide deep mechanistic insights to how embryo develops would be very hard. So this was the motivation. And I would like to show you this model system that Hannah mentioned very kindly at the beginning of the introductions that we have published now a year ago, that is based really on the same principle that we use for creating the mouse model. So we take ES cells to mimic epiblast. We take actually not stem cells for hypoblast, but we program them by overexpressing 2 transcription factors, GATA6 and SOC17, to program the cells towards this fate. And for trophoblast, we overexpress GATA3 and epi2 gamma. And we mix those cells together in a dish. And after 4 days, you see the structure that make the lumen within the central domain. We can generate those. We were trying many different pluripotency states. I'm not going to bore you with the detail to find out some of them are more amenable to make those models better than others. We found many different ratios of cells that we have to put together to make those models. But all of this is just very recently, I think just this month, or maybe last month, published protocol by Carlos that shows all of the nitty-gritties of what to do to make this model successful in your own lab. And finally, we see that this epiblast domain with time matures and start to express markers that shows that they start to change into amnion. So we can see the symmetry breaking point of epiblast of epiblast domain to form not only epiblast disc but also amnion within the same circle of cells. So we can do time-lapse movies. So we created this line, GATA3 cherry line, that would mark amnion formation to look at this process in time-lapse movies. Okay, and we can look at a role of different signaling pathways in the system. So we can, for example, look what's the role of BMP or nodal in the system and how it affects formation of different lineages. I'm not going to tell you the results because it's just boring, but it's just to tell you that this model system is amenable to studies that would not be possible otherwise with real human embryos. So at the end, I would like to show you two results. So one thing I wish to stress, though, that I wish to stress one thing, that while this model developed very well at the beginning to form this group of central cells that will be destined to form body and amnion, and those outside cells that mimic extraembryonic structures, symmetry breaking to set up the major body axis, anterior-posterior axis in this model is not occurring very well. It does occur well, but does occur very infrequently. That's what I wish to stress. So there is so much more work that have to be put towards this model to really see how it can break symmetry to make epiblast on one side, amnion on the other, and specify this group of cells that will make mesoderm on one side, so anterior-posterior axis. Okay, so I would like to show at the end just two examples of the study that one can do to use this model to understand about human embryo development. So one will be about PGCs and the other one will be about this center that makes possible to make a head. So to characterize this model, we took embryos, embryo-like structures between day 2 and 8, and for single-cell RNA-seq, and to identify all different type of cells that we can see in these models, which cell type form, which cells type do not form. I used to also stress for those of you who might actually heard or work on different models, often when we do organoids, for example, we prompt particular type of behavior by adding growth factors of specific nature to the media, for example, BMP. Here, we don't do anything like that. So everything that happens in this model is based on cell-cell natural interactions. So that's what I think is a unique feature of this model, that if you wish to understand the role of BMP or Wnt, you actually can now modulate it because you don't need these signalings to make the model as such, apart from the cell-cell interactions that naturally might provide some of that signaling. So it's like a pure tabula rasa in a sense. That's what I'm thinking, based on the cell-cell partnership. Okay, so one of the things that we've noticed when we characterize this identity of individual cells in this model, that we can see primordial germ cells. So we are able to score their presence, and indeed on day 4 we confirm by immunostaining, and they are positive for these 3 factors, that we see some of them coming in this central domain, and by day 6, so 2 days later, there are many more of them. So then we ask the question, which actually signaling is critical at that stage of those cells formation? And to address this question, we predicted that it might be BMP, because in mouse it is BMP that is important for formation of the cells. So we asked first whether the factors that respond to BMP, so whether we see phosphosmat in the right places, and IG factors, whether they come up in the right places, and they do. So then we inhibited BMP, and we found that in culture, and we found that in this case we lost PGC formation, and we also lose amnion formation. So this data altogether is now building a vision together with other model systems that exist for formation of PGCs from other stem cell derived systems, that amnion and PGCs are sharing common trajectory, and describe generated from the same progenitor cells. Okay, and finally, I've told you that I will tell you a bit about this anterior signaling center. So studying real human embryos allow us to identify the signaling centers. And again, by RNA-seq, we were able to identify which genes are present in the center. So we were able now to use the markers that will help us identify whether this center forms in the model. In the mouse embryo, in also mouse embryo model, we also see the center formation migrates towards one side, and it secretes Wnt, BMP, and nodal inhibitors to protect this part of the epiblast in its vicinity for anteriorizing signals later. So we were able to see that we can have this model making this anterior signaling center, but only if we switch off SOC17. So in our original design, we couldn't see this model because we were overexpressing GATA6 and SOC17. So of course, I'm cutting long story short, because how would we know it? We didn't actually know originally, but then when we analyze single-cell RNA-seq data from real human embryos, we started to have a hint that maybe SOC17 is antagonist of that center, SOC17 expression. And indeed, when we remove it and we induce the fate of this hypoblast type of cells with GATA6 only, we start to see this expression of genes marking the center. Oh, I don't know how. Oh, yes, I can. And it correlates very much with brachyury expression. Okay, so I'm going to finish here, just not to take too much of your time. That's mean the story number 3 about impact of environmental factors will be remaining for the future. Are you sure? Not only first row, are there other Sure? Okay, okay. So, but I would make it short. Okay, but that's I will tell you with pleasure. So just to summarize very quickly before I go there. Okay, so I've told you that how influential it was for us to break through this barrier of implantation, to give us a hint of what we should do, which type of cells and in which proportions we should put together to this dish to create these embryo-like structures that now in the mouse at least carry on development, including this anterior specification of this anterior signaling center, although we now have it also in the human, that migrates and leads to the early stages of organogenesis. In human model, we are not that far. So there are some papers, including ours, that try to push those models further, but I wish to straight away say we are not that far. Mouse model is much stronger, much better than human model. And now, of course, ethical question also exists: how far we should be able to culture safely this human embryo model. Okay, so I'm going to leave this question unanswered for now, just to tell you because of the encouragement. But if you are really bored, you should just wave or do something. Then I would not have to tell you this third story, and I will leave it for future. So you might think, okay, so we have these models. We learn a lot about basic developmental biology of how human embryos develop post-implantation and mouse too because of the models, but can we use them to something else that can be useful? Like, can we do screens for factors that often pregnant women are subjected to and check whether they matter? So I would like to show you an example of proof of principle of such a system. So for this, we actually would be mimicking, so I would tell you a little bit about another model we created, but I would just mention it rather than describe you how. So we are here trying to mimic pre-implantation stage of development by using the same type of ES cells that I've mentioned before, so Y-type ES cells, GATA4 induced ES cells, and here we add trophoblast stem cells. So this is mouse model at the moment. And when we induce GATA4 in this model, those cells change by DOX. Those cells move to form primitive endoderm. So this model is called blastoids. We are actually not the first group to create blastoids. There's other models we created first, but we were focused on post-implantation models. Blastoid model was first created by Nicolas Rivron, working in Utrecht. And it's very, very useful model for creating this pre-implantation-like stage embryo. So here we slightly modified the protocol to make it more efficient, but it is the same sort of idea that you aggregate different type of cells. Then in this case, we aggregate ES cells, Y-type ES cells induced to form GATA4, put trophoblast stem cells, and then we add DOX to be able to turn these cells into primitive endoderm. And we form the structure that you see look very similar to blastocyst. So it's a very efficient model. So we can create 1,200 blastoids per experiment per just one single agrowell plate. And from this, around 80% have this morphology. So you very comparable. So this is sort of reconstruction of how to look to natural mouse embryo at that stage. Okay, so we thought this is such a highly reproducible model, much more reproducible than post-implantation model, that reproducibility is between 20 to 30%. Again, we can make hundreds of them per day because we can start with many agrowells, with many reactions. But this model is really very, very robust with this protocol. So we thought, why wouldn't we first do a screen to check whether exposure of mothers to caffeine, ethanol, nicotine, and high and low protein diet matters at these very early stages of life. And the result was very surprising. At least I am mother twice. Two mothers is within me. How should I say? I was mother twice. No, I'm still a mother. I was pregnant twice. Gosh, I am rushing too much. I have two children. I was pregnant twice. And I did drink coffee and I did drink alcohol from time to time, particularly at pre-implantation stages of my children's pregnancy, because obviously I did not know I was pregnant. But when I will show you the results of caffeine, because protein diet is even more important, it was striking to me to see that actually, when you look at the embryo exposed to coffee, so this is equivalent to 2 cups per day, you form blastocyst. Right, so look really pretty cool, but the sorting of those cells inside these two types of inner cell mass cells that have to form either epiblast or primitive endoderm is grossly affected, and the number of cells are lower. So the development of the caffeine exposed embryo is successful but delayed. It's much worse with the high and low protein diet, particularly low protein diet. So how protein diet actually does not affect development much. Maybe it's the sorting of this red and green cells within the inner cell mass complex is slightly affected. But you can see how massively the morphology of the blastocyst stage embryo is affected if you eliminate essential amino acids. And this is striking because it is believed that pre-implantation culture does not require any specific nutritional enhancement of the environment for processes to occur normally. And actually, there were studies on mouse embryos carried out in this different environments, so we can compare our results with blastoids to real mouse embryos that were published. Okay, so here is really the end of my seminar and then acknowledgement. So I don't know what you think, but I found, my student actually who carried on this work sent me this picture, and I found this sculpture. It's not really sculpture of the embryo, but it very much can remind you of embryo. It does remind me of embryo, and it shows that we have this robust development, and yet so fragile that even apparently harmless substances like caffeine can substantially endanger the process. And I feel that symbolic nature of this little thread that carried the structure, at least in my mind, reminds me that we have to be careful when we are pregnant. Not only then, though. Okay, so just to finish, I wish to say that I talked about some of those models. So I talked about blastoids at the end and show you an example of a screen that we carried out for environmental factors, how that affect the early stages of development. I told you about post-implantation models mentioned in the mouse, only some, just this one, and some human models too. But there are many, many more models right now. This is very like playground for developmental biologists now to create these models. They're extremely ugly in comparison to real actors, like real embryos at the equivalent stages, but at the same time, they are very beautiful because they provide variety of the questions that can be challenged with them. So I don't know whether you think they're ugly. Maybe I should not say that because your children are not ugly, but you know what I mean. Like they're not perfect, right? They are not yet there, but they're already incredibly useful. And the study, all of this started only in 2014. So actually, it's only 10 years ago when we started to create these different species of stem cell-derived embryo models. Okay, the more complex they are, the less efficient they are. So that's why simple models are much more efficient in terms of reproducibility. The major technical challenge is to make human model that develops as well as the mouse, and also ask the question, how far can we go with that model? So this is really the summary. So stressing one more time that learning about pre and post-implantation development allow us to build those embryo models, and now we can use them to carry out the screens that would never be possible with real human embryos. So I think it really provides us a number of these unprecedented potentials. And therefore, I highly encourage you to include models in your research. So that's the most important slide. I've moved my lab from Cambridge to Caltech. I'm actually closing my Cambridge lab this year. It's very sad because I was there for more than 20 years, and still some people are there. Some of them moved with me to Caltech. Some of them we are looking for position to do something else. In friendship with us, but far away from us. I would like to thank all of the postdocs. I normally show their photographs on my slides, but this time I didn't, just to make slides simpler. All of the funding bodies. This doesn't mean we are currently funded by all of those people, but I wanted to acknowledge them, because if not them, we would never be able to do models, because it was very controversial, and majority of my grant applications were rejected when I wanted to apply and say honestly that I would do create stem cell models that will mimic embryos. I wish to acknowledge all of the collaborators that were extremely important for the work that I presented today. And these are ex-lab members that now have their groups, and I'm very proud of them. And these are the ones that contributed to this work. And finally, we are celebrating our human embryo lineage tracing study, the first story I've told you. And finally, I would like to thank you for listening, and lab managers in both labs. Thank you.
There's a question over here, but before we get to that question, my first one is, you moved your lab from Cambridge to Caltech over the pandemic. So that must have been quite a struggle to move during a time when it was very difficult to move around. But now you're here and settled. How are you finding science in the United States and being a scientist and a mentor in the United States compared to Cambridge?
Wow, this is a very good question. I could write a book about it. But well, I absolutely, I can't perhaps say about all of the states, because obviously I only live in California, but I love how it feels to do science here. And the difference, I mean, I love being in Cambridge too. So it was actually a hard question if I were to answer it entirely honestly. And I was saying, meeting with Catherine this morning, that California is more sunny, Cambridge is more rainy. But actually, at the end, it does not really matter. It's the matter of the people you are working with. The most challenging aspect of this move was really to establish new lab during the pandemic, if you are not on a bench yourself, because you are a bit removed from it, to recreate that momentum that we finally recreated of the discovery. This was a challenging. So if you were to be moving once your lab or setting up your new lab, you have a permission to contact me, and I can share all of different things that I have to share about it. But I feel that one big difference for human embryo study is between UK and US is that in UK, we have this day 14 rule that we have to respect when we study human embryo development. In US, it's much more variable. Different states have a different recommendations. And for example, in California, we would be not able to study human embryos beyond day 12. So that's, of course, is something that, as you know, we are trying to change, but this might take time. So this regulatory, hurdles, I think, would need to be overcome at one point if we wish to make a progress in that research.
Great. So we had a question over here first.
Hi, beautiful work. My question is on the slide that you showed between the division of the human embryo, where you identified the alpha and beta, and you identified that there are different proteins which are different in alpha and beta. I was wondering if it's hypothetically possible to separate these proteins into like a specific signaling pathway. Like you talked about the BMP pathway. Could that difference between the alpha and beta be because of like one or two signaling pathways, which are major primary pathways?
This is an excellent question, and here I have an answer, I think, and maybe not entirely satisfactory answer, but we didn't look at specific signaling pathways at these very early stages, as the signaling between cells only starts later, when they was in the human is day 5, 6 embryo, when it actually starts implanting. When we form 3 different lineages, those cells start to signal. These early blastomers do not signal so much between each other, from what we know. So we didn't look at signaling pathways at that stage. There are many of the ligands and receptors for those pathways that will not be expressed yet at that time. They only, when they start to have different characters, they become more apparent. So I think that at this early stages of development, the regulation is more based on which proteins will be degraded versus stabilized translation. That's a process of transport. These are the pathways that we see being differentially represented between those alpha and beta blastomers.
And so do you think that this is similar to other species like Drosophila or C. elegans?
Yeah, I think yes and not. So I believe that in other species, we have very strictly, for example, in Drosophila, Bicoid, Oscar, Gherkin, there are transcripts that are very well asymmetrically positioned within the oocyte. And this is what really directs the identity of those cells from the very beginning. And here we still are trying to search what these factors are, but they would not be having the same deterministic manner of action in the mouse as this is, or human as it is in Drosophila.
Thank you.
So we have time for 2 more questions. So we have one behind you, Stephanie, Hannah.
That was amazing talk, and the last part was scary, looking at the caffeinated blasters. I had a question about it, like how always thought that early that the embryo is sheltered from the maternal influence, like how much is there actual transfer from the mother, and is there a study, a mouse mother drinking caffeine? What actually happens in a real life situation?
Yeah, that's a very good point, and I also worrying about that. So there are studies. So this pilot screen that we carried out was based on actually screen of real embryos in the mouse that were the data. What we found in literature, the data was provided to be able to relate it. So actually, this discoveries that I've made maybe were not described as well because they were not studied to the same extent of detail, but there were already hints that caffeine is bad for mouse mothers.
Okay, one more question.
Thank you for the talk. It's in India. What would you think about factors in general, for example, the cell lines or the weekend crop? Particularly, you talk about the whether it's not tied to the quality of organization and static this, and then the difference in the
Yeah, so that's a very, very good question too. If it was just cell size, I would not expect to have different classes of proteins present in small or big cell, as a mirror image of each other, if you see what I mean. I would imagine that majority of the proteins that we can identify and measure at this stage would be present more in a bigger cell. This is not the case, right? There's class of proteins that it's in big cells and class of enriched in big cells and class of protein enriched in small cells. So does size matter? Well, bigger cell divides first, but I don't know whether this is because it's bigger. So, most likely it is somehow related, but I really don't have a very good answer to it. And I was trying to see in the literature whether we have situation that always bigger cell divides first, but I do not see strict evidence for that. There are some maybe I still need to search more. This finding is only few months old.
Thank you.
And you ask us about extracellular matrix, right, and mechanical input. This has enormous role in development, both of mouse and human embryo. And actually, we do study the effect of extracellular matrix on cell migration and cell fate specification. I just didn't have time to talk about this today, but yeah, it matters. So not only chemical cues, but also mechanical cues direct sulfate at that time.
Thanks.
Okay, so I'd like to end by giving 3 types of thanks. One is thank you so much, Magda, for a beautiful talk today and joining us to discuss embryos and embryo models. So please thank Magda. The second type of thanks, again, I would like to thank our co-sponsors, the Broad Stem Cell Research Center, Molecular Cell and Developmental Biology, and the Barbara Streisand Center for the Study of Women. And I'd also like to thank all of you very much for attending today and demonstrating interest in reproductive science, stem cell biology, and this interaction and integration between stem cell biology and reproductive science. Our center is funded by philanthropy and also through grant funding, so by you being here today demonstrates the importance of the topic and the importance that we place in reproductive health and science, so thank you.